Okay, the last topic covered in chapter 4 is kind of a big one, cellular respiration. We've already seen that living things need to take in energy from external sources and transform it into energy that can be used to power our cells and our body systems. We, as humans, take in the energy from the food that we eat and that contain those large macromolecules, carbohydrates, lipids, and proteins. Then we break down those macromolecules into their smaller components like glucose, fatty acids, and amino acids. We then absorb those smaller components into our bloodstream and transport them to where the energy is needed. The cell that needs the energy breaks down the chemical bonds where the energy is stored into something that it can be used to power its own processes. And this process of breaking down food into ATP is called metabolism. Cells have two ways of doing this: cellular respiration and fermentation. Cellular respiration requires oxygen to break down glucose into carbon dioxide and water and energy, ATP. And this happens in 4 steps: glycolysis, the preparatory reaction, the citric acid cycle, and the electron transport chain. We're going to go through each of those steps individually to see how that happens. So step 1 is glycolysis, and that occurs in the cytoplasm of the cell. So this is out in the goop surrounding all of the organelles, the cytoplasm. The following 3 steps are going to take place inside the mitochondria. Glycolysis literally means sugar splitting. Glyco meaning sugar, lysis meaning splitting. So the process of glycolysis is going to involve the splitting of a sugar. Glucose is a 6-carbon sugar, and through glycolysis it's split into 2 molecules of pyruvate, each having 3 carbons. Glycolysis occurs with its own set of steps that each require a specific enzyme. For the purposes of this class, you guys do not need to know all of the individual steps. If you go on to take something like A&P or an advanced biology class, you might go into a lot more detail on those individual biochemical steps. But for the purpose of this class, what I want you to know about each of these steps is what goes in and what comes out. So for step 1, glycolysis, what goes in is a molecule of glucose, and what comes out are 2 molecules of pyruvate. We're also going to get a net gain of 2 molecules of ATP through this process. But 2 molecules of ATP in the grand scheme of things is not that great. So we definitely want to go through the next 3 steps as well. The third thing that I'd like you to know about each of these steps is where it happens. So glycolysis happens in the cytoplasm. What goes in is glucose, and what comes out are 2 molecules of pyruvate. Step 2 is the preparatory reaction, or sometimes it's called the transition reaction. And this happens in the inner compartment of the mitochondrion. So now we have entered the mitochondria and we're in that inner phase. So here, the pyruvate interacts with an enzyme called coenzyme A, and it's going to remove 1 more carbon from each of those pyruvate molecules. And it uses each one of those carbons to create a molecule of carbon dioxide. The 2-carbon molecule that's left over is called the acetyl group, and that binds to coenzyme A to form the molecule acetyl-CoA. And that acetyl-CoA is the product that is shipped to the next step, the citric acid cycle. So here's the first example where we're starting to get to some of the waste products associated with cellular respiration. We're using oxygen to fuel this, but we're also creating carbon dioxide, which is something that we are going to have to get rid of as part of this process of metabolism. So this is happening in the inner compartment of the mitochondrion. What goes in is pyruvate. What comes out is acetyl-CoA and carbon dioxide, a waste product. Step 3 is the citric acid cycle. Again, this looks crazy because it happens in a series of its own 8 chemical reactions. For the purposes of this class, you are not responsible for knowing each of those individual chemical reactions. But, kind of as an overview, acetyl-CoA reacts with a 4-carbon compound and then completes the cycle of those 8 chemical reactions. And this loop of reactions is actually called the Krebs cycle. Each acetyl-CoA molecule that enters the cycle will produce 2 molecules of ATP and some nicotine adenine dinucleotide, that NADH, and some FADH. We saw some of these molecules produced after some of the previous steps also, but these are important because these, NADH and FADH2, are what is going to build up and fuel the next step, which is going to be the electron transport chain. Again, this is happening in the inner compartment of the mitochondrion. So as we go and each of those acetyl-CoA molecules is going through the Krebs cycle, we're building up a concentration of NADH and FADH2. That's important. We're building up the concentration. We're creating a concentration gradient, if you will, which will lead us to the final step, which is the electron transport chain. And this is what's happening in all of those many folds of the inner membrane of the mitochondrion. So when we were looking at the picture of the mitochondrion before, that inner membrane was highly folded. We need all of that surface area to be able to fuel the electron transport chain. Those molecules of NADH and FADH2 produced in those first 3 steps are then passed along a series of carrier proteins that are embedded in this inner membrane. And their molecules pass their electrons to each of these proteins, and each time an electron is transferred, energy is released as ATP. So for each molecule of glucose that's split by glycolysis, we are then going to get 32 molecules of ATP once we get to the electron transport chain. That is a very big return on investment. So 32 molecules from only 1 molecule of glucose. So every glucose that we split off of one of those larger complex carbohydrates, we're going to get 32 molecules of ATP out of the electron transport chain. Hopefully, after watching all 4 of these videos, you have answered all of these questions. If you still have questions about any of these topics, please feel free to email me. I know it's much harder to cover everything in this video format than if we were having this discussion in class. So please do not feel that you are all on your own. You can always email me or set up a time to talk on Zoom or Teams if you want to go over any of these in more detail. I'm always happy to do that.